flu. And I'll talk about the vaccine a little bit later when we get into the influenza virus. But first I want to tell you a little bit about the basic biology of the virus, because I need to do that to get to talk a little bit about how why the vaccine works the way it does.
But unlike the cold,
when you look at an electron micrograph of an influenza virus particle, if you remember that coronavirus, there's that kind of halo around it. Influenza viruses have that as well. It's not as easy to see in this picture, but you see there are little projections that stick out from the viral particle. And those are the surface proteins of the influenza virus. And there are two main proteins that are on the surface of an influenza virus particle. The first is a molecule called hemagglutinin. That makes up about 80% of the viral surface proteins. And the hemagglutinin is also shortened to H, and you'll see that. The second protein is neuraminidase, or N, and they make up the bulk of the surface proteins. There's also a small amount of a protein called M2 on the surface. It's an ion channel. Okay? So there are three surface proteins on an influenza virus particle, and there are 8 segments of RNA within the virus. In those 8 segments, 6 of those segments encode for one protein, and segments 7 and 8 encode for 2 proteins. So there's a total of 10 proteins on 8 segments. And just to remind you, you see each segment is converted into mRNA, and then the mRNA is translated into protein, because remember, this is a negative sense RNA virus. So 8 segments, 10 proteins is what makes up influenza virus. And the influenza virus primarily infects the ciliated epithelial cells of the respiratory tract. And if you'll recall, the respiratory tract has those innate defenses, that layer of mucus which traps particles, and then the ciliated cells move those particles up the respiratory tract. What the influenza virus does is it infects those epithelial cells, those ciliated cells. And like any good virus, the influenza virus basically destroys those cells. So as it's coming out, it kills those ciliated cells in the respiratory tract. And that's where the secondary infections come in. Part of your defense mechanism, right, is that layer of mucus which absorbs bacteria and fungal particles that might be trying to get into your lungs. The ciliated cells move those out. If the if the influenza virus destroys those ciliated cells, your respiratory tract can't get rid of those bacterial particles. And so part of your defense mechanisms are weakened, and those bacterial particles can go on to cause pneumonia in some cases. Okay, so it's the destruction of those ciliated cells in your respiratory tract leads to the increased likelihood that you will be susceptible to bacterial infections, because those defense mechanisms that you have aren't working temporarily while those cells are being destroyed. Eventually those cells will be regenerated, but during the period of infection you have a weakened defense mechanism against invasion by other respiratory pathogens. And that's what happens with those who are immunocompromised. They become more susceptible to these secondary infections. When you become infected with an influenza virus particle, your body produces antibodies against the hemagglutinin, so the anti-H antibodies, and against the neuraminidase, anti-N antibodies. The hemagglutinin antibodies bind and neutralize the virus, so they prevent the virus from getting into cells, and the neuraminidase antibodies prevent the virus from spreading from one cell to another. So the hemagglutinin and neuraminidase molecules are really important in terms of your immune system's ability to fight and recognize influenza virus infections. So the majority of your immune system's adapted defenses are based on the humoral immune response targeting the hemagglutinin and neuraminidase molecules. The reason why you become infected year after year, or can potentially become infected year after year by influenza virus, is because the virus keeps on changing. Okay. In most years, it's a subtle less dramatic change. So you have a change in amino acids on the hemagglutinin and neuraminidase molecules. If you get the right amino acid changes, antibodies that your body had used to protect you previously will no longer recognize the hemagglutinin or neuraminidase in the new influenza virus strain. And so you can become infected with the virus. Okay. So as long as you have antibodies against the current circulating type of influenza virus, you won't get the flu. And that's why in most cases the vaccine works. You get vaccinated against a flu strain, you raise hemagglutinin and neuraminidase antibodies that protect you from infection. But over time, the virus changes. Mutations occur, amino acids change of the hemagglutinin and neuraminidase molecule, and those antibodies no longer protect you, and you can become susceptible to infection. Now, to complicate things even more, there are at least 16 different types of hemagglutinin molecules and 9 different types of neuraminidase molecules. Okay, and that collection of hemagglutinin and neuraminidase is found in birds. A subset of these hemagglutinin and neuraminidase molecules are also found in human circulating influenza viruses. Okay. The virus itself is named based on the type of hemagglutinin and neuraminidase molecule it has. So, for example, bird flu is an H5N1. It has hemagglutinin subtype 5, neuraminidase subtype 1. So when you hear an H1N1 or an H3N2, it's referring to the subtype of hemagglutinin and neuraminidase that make up that particular virus particle. Now, influenza virus is really a bird virus. It's found in aquatic species of birds. And what you see in humans is when these particular influenza viruses cross over and infect humans. So the hemagglutinin and neuraminidase that we have circulating in human viruses are derived from these bird influenza viruses. Okay. So influenza is really a bird virus, and occasionally those bird viruses acquire the ability to replicate in human cells, and we'll talk about that shortly. So each subtype, H1 and H2, for example, differ from each other. Those hemagglutinin molecules differ by about 30% at the amino acid level. So it's still a hemagglutinin molecule, but they are fairly divergent, so only about 70% identical at the amino acid level. Okay. And only 6 subtypes of hemagglutinins have been found in viruses that infect humans. That's H1, H2, H3, H5, H7, and H9. But really only H1, H2, and H3 are in influenza viruses that spread between humans. H5, H7, and H9 have been found in humans that have become infected with influenza virus, but they've acquired those infections directly from birds. And the virus doesn't spread from one human to another. Okay, so although H5, H7, and H9 influenza virus infections have been found in humans, they've only been acquired through coming into contact with infected birds. There's no human-to-human transmission of those influenza viruses. Now, because influenza viruses are constantly changing, there's a fairly sophisticated naming system that allows scientists to basically differentiate between different strains of influenza virus. So the naming convention is the influenza virus is type A, B, or C. The species that it's isolated from, if it's from humans, we just drop that. The place of isolation, the strain designation, which is usually let's say it's the 15th influenza virus that we've isolated from Peterborough, then that's strain 15, the year that it's isolated, and the H and the N subtype. So to give you an example, in influenza A, isolated from the first patient in Hong Kong in 1968, is given the name A Hong Kong/1, because it's the first strain that was isolated. 68's the year, and H3N2. is the type of hemagglutinin and neuraminidase in it. Okay, so that tells you a little bit about that particular strain. And so that's the scientific naming for influenza viruses. Now I want to talk a little bit about the replication cycle of influenza virus, and I gave you a broad overview of viral replication cycles, but this is a good chance to review that, but we also need to sort of put in the replication cycle to understand a little bit about how pandemics occur and also how the antiviral drugs that we can use fit into this picture. So the first step in entry is binding to a cell surface receptor, and it's that hemagglutinin molecule, or H molecule, which binds to sialic acid residues on the surface of cells. Sialic acid is a carbohydrate. It's part of glycoproteins or glycosylation of proteins. And so every cell has these sialic acid residues attached to surface proteins. Once bound, the virus is endocytosed. And when it's in the endosome, the low pH initiates a conformational change in that hemagglutinin molecule, so the chains within it rearrange, which promotes fusion between the viral and the endosomal membrane. As the fusion process is going on, that M2 ion channel, which is on the surface of the viral particle, hydrogen ions are transported inside the viral particle, and that releases the viral RNA, which is normally bound to the capsid. So without the M2 ion channel, the virus can't uncoat. So the viral RNA will remain stuck inside the virus. Okay? Once the RNA leaves the viral particle, it's transported into the nucleus of the cell. And I'll point out that, again, the nucleus is an odd place for an RNA virus to replicate. And this is the only RNA virus I think that we'll talk about in class that actually replicates in the nucleus of the cell. All the other RNA viruses that we'll be discussing replicate in the cytoplasm. And I told you why it's important for most RNA viruses to replicate in the cytoplasm. But flu viruses are the exception. In the positive sense, single-stranded RNA viruses, we talked about the idea of translational control. How do these viruses interact with the host translation machinery? And we saw things like IRESs and picornaviruses, or the VPG protein and caliciviruses, as ways that the virus gets around that 5' cap that it has to deal with. The orthomyxoviruses use what's called a cap-snatching mechanism. And essentially what happens is the virus steals the cap off of a cellular RNA. So in the nucleus of the cell, your cell makes mRNA, and that mRNA has a 5' cap on it. Okay. The viral polymerase binds to that viral mRNA, or cellular mRNA, about 15 nucleotides in. The polymerase cleaves that cellular RNA and takes the cap plus the first 15 nucleotides. It then takes that cap, moves down to a piece of viral RNA, and then uses this as a primer to essentially transcribe a complementary copy of RNA. So at the end of this replication process, the virus has taken a cellular cap and attached it to a piece of viral RNA. Okay. This capped RNA can then be exported from the nucleus and translated into a protein. Okay, a process known as cap snatching. So rather than having IRESs or VPGs, just takes a cellular RNA, mRNA, cuts the cap off, uses that to prime its own transcription of its own RNA. Now the RNA has a 5' cap, it can be translated into protein. So in the nucleus, I'll just skip over, but the viral RNA is replicated, viral proteins are produced, and then you get assembly of the viral particle, which happens either in the cytoplasm or the ER. And one